Is my voice clear?
Sir. Good morning, sir.
Pavitra ma'am, are you there?
Yes, ma'am. Yes.
Okay. Okay, sir. I will start. A warm welcome to all of you from Telangana Social Welfare Residential Degree College for Women, LB Nagar, Hyderabad. I am Dr. D. Kavita, HOD, Department of Botany. Department of Botany organizing one-day national webinar on forensic importance of pollen. To start the session, now I welcome Chairperson of this webinar, Dr. B. Arundhati Garu, Principal, Telangana Social Welfare Residential Degree College for Women, LB Nagar, Hyderabad. Due to some reasons, Madam could not join this meeting. Maybe she will join in the last. Next, I welcome Vice Principal, Dr. Pavitravani Karsha, HOD, Department of Microbiology. Welcome you, Madam. Next, I welcome our today's speaker, Dr. A. Vijayabhaskar Reddy Garu, Assistant Professor, Applied Palynology Laboratory, Department of Botany, Usmania University, Hyderabad. Thank you, sir, for accepting our invitation. Due to some reasons, we couldn't conduct this webinar as per the schedule, even though you have given your valuable time for us. Once again, thank you and welcome you, sir. Finally, I welcome all faculty members, my colleagues, research scholars and students who are attending this webinar. Now I request our Vice Principal, ma'am, Dr. Pavitravani Karsha, to address the session. Over to madam.
Thank you, Dr. Kavita, madam. Good morning and a very warm welcome to each and everyone to this one-day national webinar on forensic importance of pollen. I specially welcome guest speaker of today's webinar, Dr. A. Vijayabhaskar Reddy Garu, Assistant Professor, Applied Palynology Laboratory, Department of Botany, Usmania University, Hyderabad. Telangana Social Welfare Residential Educational Institutions is running 268 residential educational institutions from 5th standard to UG and PG level. The government of Telangana has sanctioned 30 residential degree colleges in the year 2016 with a vision to build an outstanding educational institutions which provide high quality higher education, holistic and value-based education to the marginalized children on par with the other advantaged children in the world. The mission of our educational institutions is to prepare the students to face the challenges of the 21st century by providing a wide range of learning opportunities in classroom and beyond to help the students realize their full potential. Our college is offering 7 courses at BSc, BCom, BA with 840 students intake. We have 36 well-qualified and experienced staff working in our college. During this pandemic, we have not stopped the teaching learning process. We reached the students through various online platforms and also through village learning centers, a new experiment initiated by our Honorable Secretary Dr. R.S. Praveen Kumar IPS, which is recognized globally. One such platform is webinars. Our college conducted 20 webinars in various subjects in the last 4 months. At this moment, I thank our Honorable Secretary Sir, Joint Secretary Sir, ZCO and OSDs being leaders and the catalyst that inspire us to do the best. And also our Principal Madam for encouraging us all the time. I congratulate Kavita, Department of Botany, for organizing this webinar. With these few words, I welcome once again our speaker, Dr. Vijayabhaskar Reddy Garu, faculty, students, and all other participants to this webinar. Thank you. Thank you so much. Over to Kavita.
Thank you, ma'am. Really, it was a wonderful introduction about our institutions. And now I'm taking privilege to introduce our today's speaker, Dr. A. Vijayabhaskar Reddy, sir. Sir was born in Mettapalli village, Suryapet district, Telangana state in 1976. He received MS degree and a PhD in PhD degree in botany from Osmania University. He joined Osmania University in 2013, where he supervises research activities in the field of applied palynology. He currently serves as head Department of Botany in Nizam College. His research is based on the field of applied palynology with a special focus on palynotaxonomy, melisopaleontology, clinical palynology, and forensic palynology. He teaches several courses on plant taxonomy, palynology, and biodiversity to bachelor students and master students. He also organized many national workshops, seminars, and conferences for academicians and professionals in collaboration with national funding agencies like UGC, NMPB, etc. He has given several invited talks at national seminars and conferences. He is also author and co-author of 43 research papers in national and international referred journals and more than 10 conferences contribution, 5 books and 1 patent. He is the life member of many professional bodies and in 2016 he was awarded the Fellow of Scientific Society of Advanced Research and Social Change. Now I request Vijay Bhaskar Reddy sir to take over the session. Over to sir.
Thank you, Dr. Kavita, for a nice introduction. Am I audible?
Yes, sir. Yes, sir. Please go ahead.
Yeah. Also thank and congratulate the organizers, Dr. R.S. Praveen Kumar, IPS Secretary, and Dr. Praveen Mamidala, Joint Secretary, who's also my colleague when we joined in the university, same time, and the Chief Advisors, Dr. P. Bhanu Prasad and Dr. Rajni, and the Chief Chairperson of this webinar, Dr. A. V. Arundhati, and Dr. Kavita for giving me this opportunity. Before going to this, I'll start my presentation.
Swati madam, kindly make a host Vijay Bhaskar Reddy sir.
Okay, madam.
Yeah.
Thank you.
Please enable.
One.
Done, sir.
Yes, yes. Yeah. Is it visible?
Yes, sir.
Yeah. Fine. Forensic palynology. Before going into the details of the forensic palynology, I would like to introduce you a forensic science. What is a forensic science? This word has come from the Latin adjective that's forensis. The exact meaning of that forensis is before the forum, because in ancient days, particularly in Roman times, criminal charge meant presenting the case before a group of public individuals in the forum. So that's why the term forensic is effectively a synonym for the legal or related to the courts. They have chosen this Latin word and for this subject called forensic science. Forensic science is nothing but it is an application of the tools of science as a specific scientific facts to help or solve the all legal issues. The main aim of the forensic scientist is to establish or to determination of the cause, a location, and the time of the death for an incident. With this goal, a forensic scientist work and solve the different legal issues or crimes. Yeah. Let us explore the history of the forensic science. There are many eminent personalities with their efforts. This forensic science have become emerged to a complete subject with many applied branches. So here, these two are persons, Sir Arthur Conan Doyle and Sherlock Holmes series. These two contributed a lot to the forensic science. Particularly, the Holmes series introduced many ideas that are now major parts of forensic science, like serology, document analysis, firearm identification, and much more. And also, the Eureka movement is considered to be the earliest account of forensic science. King ordered the Archimedes to find out the crown, which was made up of gold, and he had a doubt whether that was exactly or purely made up of gold or not, and that assignment was given to the Archimedes. And Archimedes was thinking about yes. Yeah, Archimedes was thinking about that that assignment, and while taking a bath, that some water was displaced when he gone into the tub, and that gave him an idea. And this he used this water displacement and proved that the king's crown was not made of solid gold by its density and buoyancy. So this was the first attempt in forensic science. Since then, there were many developments and many fields were emerged. And fingerprinting, of course, was the first application of the forensic science by the Chinese, who used these fingerprints to identify various documents and clay sculptures in the 1700s. Of course, they didn't have a proper classification system, but they used these fingerprints and for the evidence to solve the various legal aspects. And these are various people who contributed a lot to the development of this science. And there are many branches, many applied aspects of the forensic science, and each person has his own contribution in the development of these applied aspects. Bernard Spilsbury is said to be a father of forensic science, and father of modern forensic science. Credit goes to the Dr. Edmund Locard, and father of ballistics, Calvin Hooker Goddard, and Carl Landsteiner is the father of blood group identification. Still, in many cases, this is a very important ingredient in solving of the crime. And of course, Arthur Koehler, he has a credit taken, and his work in the particular field of botany, application of botanical aspects in solving of the crime, emerged as a forensic botany. He is too called as a father of forensic botany. And the father of criminology, credit goes to the Cesare Lombroso, and father of computer forensics. Nowadays, most of the fields are connected, automated with the computers, and computers are playing an important role in every field of human life. So because of hacking and other aspects, this has emerged as a separate science, that is a father computer forensics, and Michael Anderson said to be father of computer forensics. And what we are going to discuss today, forensic criminology, is the one brand. Credit goes to this person who is working in Texas University, said to be a father of forensic criminology. He solved a number of cases just by using teeny microscopic particles that are pollen and spores. And there are different principles, important 7 basic principles are there, entire forensic science works with. Law of individuality, principle of exchange, law of progressive change, law of comparison, law of analysis, law of probability, and law of circumstantial facts. So law of individuality means here the every object, natural or man-made, whatever it is, it has an individuality. So for if you see each and every person is unique. His fingerprints are unique. No one fingerprints are matched with the other person. So this law says that individuality says that each and every person or whatever the thing available in this world is unique in one way or other way. So this gives a very important evidence in the solving of crime. And next one is a principle of exchange. According to this, when a criminal or his instrument of a crime come into contact with the victim or the object surrounding him, they leave a traces, and the criminal or his instrument picks up traces from the same contact. So this is also very important principle, very important law, where a criminal exchange his traces to the crime scene, or else he also takes some traces from the crime locality. Law for progressive change, of course, if identification or detection of crime takes a delay, there is a change in the evidences, what exactly laid down there. So time to time, there is a change. Because of that change, there is a time delay. Automatically, we lose some evidences over a period of time. So, and one important thing is, one interesting thing is here, pollen and spores. Now there is a time delay, but at any time, after many years also, many of the pollen grains can retain their shape, their size as it is. So we can work on the pollen and the spores. That's the beauty of the pollen and the spores. And law of comparison says that only the likes can be compared. It is the law of comparison. It emphasizes the necessity of providing like sample and a specimen for a comparison. We always should collect the control sample and evidence sample. From crime locality, we should collect the evidence sample, and from surrounding premises, we should collect the control sample, and we must make a comparison whether they are matching or not. If they are not matching, means we'll come to some conclusions that this law says about law of comparison. And the law of analysis, the analysis can be no better than the sample analyzed. Now the evidence is facts are recovered and collected properly. And finally, the analysis is very important. A forensic scientist should analyze the evidences, facts properly. Otherwise, he cannot establish that crime or that legal aspects properly. And the law of probability, all the identifications, definite or indefinite, are made consciously or unconsciously on the basis of probability. And law of circumstantial facts says that facts do not lie. Man can and do. Hence, the importance of a circumstantial evidence is as good as the oral evidence. So, a forensic palynologist or a forensic scientist should always depends on the circumstantial facts. He must provide the evidences from the crime locality or legal sites. Then only he can come out with the proper evidences, and he can win that or that can be solved properly. Otherwise, we cannot depend on the oral evidences. And here that Sherlock Holmes principles, this is a law of every contact by a criminal leaves behind it trace. This principle of exchange is given by this Sherlock Holmes. Forensic science is not a simple subject. It is connected with the multi subjects. It's a multidisciplinary science where it has connections with the anatomy, maths, language arts, social studies, chemistry, physical sciences, earth science, and general sciences, technology, physics, and biology. So, forensic scientist sometimes uh, uh, he works his own, and wherever possible, he depends on the different fields of these sciences, other fields of the sciences, time to time, so that he can get probable evidences, substantial evidences, and provide it in front of the court. And forensic botany, it is another branch of forensic science. It also has much importance. And there are different branches of botany, again plays an important role, like ecology, molecular biology, dendrochronology. Dendrochronology is nothing but it is a confirmation of the age of plants just by counting the annual rings. And anatomy, limnology, systematics, plant systematics, and palynology. And let us see how palynology of botany works in the solving of crimes. So, for application of palynology, particularly in the knowledge of pollen grains and as well as spores. The study of pollen and spores is called as palynology. We apply the knowledge of this pollen and spores in the solving of legal aspects or a crime is called as forensic palynology. It is a first this forensic palynology based on the palynological evidence. First case was solved in the year 1959. This is the murder case first solved by using the palynology. So soil washed from the clothing of a murdered woman. She was murdered in Sweden, and the clothes were washed, and that liquid was sent to the palynologist, and they extracted the pollen from the liquid which was taken from the washed clothes of these women, and they identified this pollen. This is the pollen. Actually, this is a Romex pollen, which was identified at the crime site, from the clothes of these women. And when compared with the control samples in surrounding samples, this plant was not appear there. This is not part of that scene, but this is attached to the clothes of this woman. That shows this murder was not taken place exactly in that site. She was murdered some other place and dumped here. And later on, this case was solved based on that evidence given provided by the palynologist. So this was the first case solved by the using palynology. And what is the palynology here? Palynology is nothing but it is a study of pollen and as well as spores. You all know the pollen and the spores are reproductive structures of plants, and pollen are produced by the higher plants like gymnosperms and angiosperms, whereas the spores are produced by the thallophytes, bryophytes, and pteridophytes. And you know, the difference between, what is the major difference between a spore and the pollen grains? And the spores which are produced by the thallophytes, bryophytes, and pteridophytes, these are having the particularly in thallophytes, these may be sexual or asexual spores, sexual and asexual spores. Whereas in bryophytes and pteridophytes, after germination, these spores produce archegonia and antheridia, and after fertilization, they produce the diploid zygote. Whereas pollen grains are reduced male gametophytes and fertilize the ovum. The main target of the pollen is to fertilize the ovule, which is totally embedded inside the ovary. So spores mostly have a monolith and trilith mark. These are the apertural marks. Apertural marks are they are also called as germ pores. where in fungal spores, from that point, the mycelia comes out and grow. In pollen grains, the particular point where the pollen tube comes out, that point is called as a pore, germ pore. That may be called as a scientific term in palynology we use as a aperture. So these apertures are various shapes, sizes, and they help in identification of the plants properly. And the variation of apertures may be a colpus, porous, colporate, and like this. And spores generally they germinate from the proximal side. There are two sides spores and pollen have, proximal side and distal side. When pollen grains are sexual spores produced by the meiotic division, each meiotic division leads to the formation of 4 cells, and they combine into a tetrad. So the side towards the center of the tetrad is called as a proximal side. The side away from the center of tetrad is called as a distal side. From the proximal side, these spores get germination, and whereas from the distal side, these pollen grains germinates. And another important thing is here, spores have wide ecological niche. Spores have wide ecological niche for germination. Wherever they get a suitable media, just they germinate. Just you see, if you place a bread at your desk or on open place and observe it for 2 or 3 days, definitely after third day or fourth day, you will observe a fungus like a mucar or else rhizopus growing on it. A bread mold grows on that. From where it has come? That means spores are omnipresent, everywhere they are present. Whenever they get a suitable media, immediately they start germination. This is the capacity of the spores. That means they have wide ecological niche. Wherever they go, they get a suitable media, they germinate. But the pollen, it cannot. It has to, no other choice, but it has to germinate only on the stigmatic surface of its own flower or else other flower of its own plant. That's a highly restriction. So these are the major differences between the spore and a pollen. And this is what you're seeing is here the pollen grain. Of course, different fields works on this. Embryologist works on this, and they show mainly interest on germination aspects of this pollen grain. How pollen germinates, from where the pollen tube comes out. This is called a germ pore, aperture region. From this region, pollen tube comes out. Once it is landed over the stigmatic surface, it produces a pollen tube. So embryologist shows interest on that aspect, whereas a cytologist shows interest only inner contents of the pollen grain. Okay, what type of what number of chromosomes present inside, and what are the other organelles, and all these aspects studied by the cytologist. Whereas the palynologist interest is only on this outermost layer called exine. This gives us a very important clues in identification of the plants. So this exine, the outermost layer, which is having various patterns, they are called sculptural aspects, ornamentation patterns, that gives a proper identification of the plant. Just by looking at a tiny structure, a pollen grain, we can identify family of a particular plant, genus of that particular plant, and even up to species level also it is possible to identify. So this is all comes under the palynology. So, and the outermost layer is made up of a tough rigid layer that is called as, that is a biopolymer called as a sporopollenin. This is outermost layer. Exine is made up of a sporopollenin, and this is inner layer. This is called as a intine. This is a very soft layer, and this exine is made up of this biopolymer sporopollenin. Due to this sporopollenin, these pollen grains, even in spores also, this sporopollenin is present, and this sporopollenin gives the pollen grains and spores a tough and a rigidity, so that they can remain for a long years. They can remain for a long years if inoxic conditions available. Even oxic conditions are available, some of the pollen grains cannot be destroyed easily by various physical and biological and even chemical activities. That makes the unique for the pollen grains, and even they are available in fossil form also. The study of those fossils is called as a paleobotany. And based on that paleopalynology, based on the paleopalynological evidences, we can reconstruct the past vegetation patterns, and even we can assume the past environmental conditions, and we can establish some facts based on these pollen evidences. And that also helps in the forensic science. And why pollen variation? Why pollen variation? See here, pollen grains generally they vary in pollen units. Pollen units are nothing but once pollen grains are formed inside the anther lobe. So this is a flower of an angiospermic flower, where you can see two essential parts. This is the androecium and this is the gynoecium, and other two parts, non-essential parts, calyx and corolla. So pollen grains generally produced in this anther lobe. Once they get a maturity, after formation, meiotic division, they generally united into tetrads, and they liberates out from the anther lobe. Once they get a maturity, either they get separated from the tetrad and single units comes out, or else as it is tetrad or sometimes some other divisions takes place, and united more of pollen grains and comes out in the form of polyads. That's polyads are important for Mimosaceae family. Majority of the Mimosaceae members produce or release the polyads. Yes, any doubt, Kalyani? Someone raised a hand. Shall I continue? Have any doubt?
I think by
I think so. I'm sorry.
What is it? Shall I continue?
Yes, sir. Yes, sir.
Yeah,
Please.
fine. Okay. Pollen units and polarity, size and shape. There are different sizes of pollen grains. You can see here different sizes they are in, and the shapes also varies from pollen grain to pollen grain, and symmetry, apertures, and exine stratification. Even outermost layer exine also shows various patterns. Some are very smooth, some are spiny, some are reticulate, and some are spiny reticulate. Like this, there are so much of variations which help in identification of the plants. And why this variation generally? See, plants produce these pollen grains, and the main target of the pollen grain is to fertilize the ovule, which is embedded inside the ovary. So once they landed over the stigmatic surface, which was taken from the anther to the stigmatic surface, that's called as a pollination. There are some mediators like different vectors involved here. They are called as a pollen vectors, birds or animals, whichever. That's again plant specific, and comes here. Example, just you assume this is a Azadirachta indica flower, and it receives because always the stigma is open to the air. It receives Azadirachta indica pollen also, as well other pollen. Tamarindus indica pollen also may come, or else any nearby plant pollen also comes and land over here. But thing is, stigma has to play a key role and allows only its own pollen grains for germination, not other pollen. For this purpose, each and every plant produces a unique type of pollen grain that varies in all these aspects, and also the chemical nature of this stigma, the exudates and other aspects also play an important role in the identification of its own pollen grain and allow it for a germination. For this, each and every plant produce a unique type of pollen grains that helps in the identification or even solving of the crime. There are anemophilous plants and zoophilous plants. Generally, plants choose based on the vectors. Plants are divided into anemophilous and zoophilous. Anemophilous plants, they are called as wind-loving plants. Generally, their pollen is transported or taken up by or pollinated by the wind. So here, these pollen grains or these plants always produce pollen grains which are lighter in weight, so that they can wind can easily take in those pollen grains out, and small in size, smooth in nature, and they produce high amount of pollen grains, always high amount of pollen grains. The pinus pollen, which produce millions of pollen grains per year. Even rumex also produce millions and millions of pollen grains per year. Within a year, you can see that amount of pollen grains. They all fall, only few of them go and fertilize, and remaining all float in the air, and because of gravity, they settle down. And they fall on various objects, and these play very important role in solving of the or detection of the crime. And dispersal also, sometimes they travel kilometers, thousands of hundreds of kilometers they travel along with the wind speed. And a forensic penologist should have idea while taking the evidence samples. He must have idea which is Anemophilus pollen and which is Entomophilus pollen, because in any evidences it traced Anemophilus plants, means though the plant is not part of that locality, there's a chance of these pollen comes from the elsewhere because of the wind velocity and wind speed. Even some pollen cross the continents also. Yeah, these are zoophilous plants, animal-loving plants dispersed by the bats, birds, bees, and etc. etc. Generally, they produce a low amount of pollen grains. They don't require, because here directly these pollinators taking the pollen grains to the stigma, so they don't require huge amount of pollen grains, and they release a few less amount of pollen grains. And they always abundant near to the plant, and even they are abundant in the nest of animal pollinators, even bee comb, honeycomb, or nest of the pollinators of birds' nest also you can find a huge amount of the pollen grains. They always matters to solve some aspects. And they dispersed in clumps to ensure fertilization. Generally, these pollen grains are always in clumps because of presence of that vicin threads. They promote the clumping, or else the tapetum, which is part of the anther lobe, which is having the plastids, they secrete the viscose exudates, viscose secretions. They forms the a pollen kit, which is the outermost layer of that exane wall. And due to this pollen kit, all these pollen grains aggregated and attached to the body parts of the insects like this. Even these all attached to the body parts of that various places, various instruments, various aspects, various people in different parts. So they always help in the identification or solving of the crime or detection of the crime. See here, this is the fall of pollen grains in different areas. Pine forest, 20,000 to 1 lakh pollen grains per centimeters square per year. Deciduous forest has this much, 5,000 to 10,000. Steppe grassland, 1,000 to 15,000. Tundra, 50 to 2,000. And desert, 50 to 20,000. These are all play important role. A penologist, a forensic penologist should have idea how much fall of these pollen grains taking place, and which is the important one to provide as the evidence. And this marijuana, it's nothing but cannabis sativa, rumex, eucalyptus. These are also produce very small and lightweight pollen grains, but falling rate is very high, nearly 2 centimeters, about 2 centimeters per second, about 2 centimeters per second. Just like a powdery dust can be seen on the all from the eucalyptus plant nearby. Any vehicle is there, or any instrument is there, any person is there, immediately they fall on that objects or these vehicles or whatever it is there just near to the plant. So these are all can be taken into consideration. And philological aspects also play an important role in the identification of the or solving of the crime. We can exactly confirm when the crime was taken place exactly, or we can assume during the particular season it was occurred. We can assume because based on these phenological aspects, each plant has its own phenology, So based on that, we can confirm the season of that crime when it was occurred. So this is a plant called Ambrosia artemisifolia. This is a common rugweed which grows mainly in America. There it causes a lot of allergies also. And generally, it remain dormancy during these periods, December, January, February. And it starts germination during the month of March. And it comes to the vegetative growth up to July, and starts flowering and releasing the pollen grains from August to November. And then it releases the seeds, and they remain in a dormant state. See, any crime took place, and though in that crime area, crime locality is identified with the pollen of these ambrosia, we can confirm that that instant was taken place during these months, August, September, October, November, because only these months we can see the pollen of this plant, and other months you don't find. So with this, we can confirm the exact month or exact season of that crime taken place based on these evidences. So what forensic palynology can do, how it is useful in the detection of a crime. So determine the travel history of items or persons. Determine the travel history of the items or persons. See, one person is traveling from the Kashmir to Kanyakumari. So while traveling from Kashmir to Kanyakumari, he also holds his clothes or his hair or other parts may trap the pollen grains. So by analyzing the person or even vehicle, we can trace out the roots of that person exactly from where that person has come from. So we can know the travel history of that item or a person and provide exact information of the environment an item has come from. So we can confirm whether the particular person has come from that locality or not. See, there are many ecosystems like a desert ecosystem, grassland ecosystem, forest ecosystem. If a person have come from the forest locality, the pollen is exactly matched with the forest. So we can say that we can confirm that the person has come from the forest or agriculture fields. The person has come from. Then the cultivated plants pollen can be reflected on the clothes of that particular person. Like this, we can get the information from where the person has come, from which environment the person has come from, and determine the body, if body was moved or shifted. So first case was solved, that was in by the Rumex pollen. So Rumex pollen was not part of that area. The woman was murdered somewhere else and dumped that body somewhere else, some other place. So the body is not part of that area. Based on that that Rumex pollen, they identified, because Rumex is not part of that. Even that's nearby also, that plant is not growing. So like this, we can confirm that determination of we can determine if body was moved and determine the season in which crime was. Yes. Yes. Someone is talking. Please mute yourself.
Why, Shirisha, kindly mute your microphone.
Someone is talking. Please mute yourself. Yeah. Determine the season in which a crime was committed. Just now I told you, based on the phenological aspects, any pollen is identified there, and based on that we can assume the exact season of that, and based on that we can confirm the season of that crime, and determine the origin or travel history of items like drugs, weapons, money, explosive devices, vehicles, and etc. And all these based on these physiological aspects, and determine the geographic contents or origin of food products. See, nowadays most advertisements you heard about that Kashmir honey, lion honey, Kashmir honey. They give advertisement. So just take out the honey, what they advertised, and analyze the sample. If you get a pollen of that Kashmir locality, then we can confirm that this is from a Kashmir locality. Otherwise, it cannot. So like this, we can confirm the origin of food products. That helps in the solving of different aspects. And much more things here. The what makes pollen so useful? Why we use the pollen for the detection of the crime or solving the different legal aspects? There are about 500,000 plants that produce a pollen or spores. Each species is unique. Already we discussed. They are unique in the different aspects. The size is plays an important role. Different sizes of pollen grains. Just they start from the 5 millimicrons to the 200. Maximum is 200 millimicrons. They're very teeny, and generally goes unnoticed. Any person, culprit or a criminal, goes to the that area, crime locality, and do the crime. During that time, he takes many precautions, but he, because of unnoticed pollen grains, he cannot take care of that pollen grains, and those pollen grains can be put for evidence to solve such type of crimes. In majority cases, it happens, and suspect picks up evidence of his or her presence at a crime scene without realizing it. Therefore, without taking precautions, he cannot rely on that. He cannot see also. See what many of you. Somebody sit in your classroom or somebody sit at home watching this. Your environment is not also free from all these spores and pollen grains. It's full of pollen and spores, and these are all gives you clues. These are all majority of we may not observe these, but they become very good evidence for the solving of the crime. Variable morphology and durability. I told you, because of presence of the that sporopollen, the outermost layer exine is made up of a sporopollen that helps the pollen stay longer period, because it's not easily digested by our biological system and destroyed by the chemical and physical activities. It stay for a long time. And each geographical region produces a unique pollen print that can be very useful in linking things to the exact location. I told you, you go to the crop fields, that agriculture ecosystem is different, forest ecosystem is different. Xeric plants, they grow in particular desert environment, that pattern is different. The pollen print for that particular geographical locality is different. Based on that, we can segregate. Rangareddy flora is different from the Khammam, or Telangana flora is different from the Andhra, or Andhra flora is different from the Kashmir. So like this, each geographical locality is having its own pollen print. Based on that, we can confirm the localities. We can take these evidences in solving of the different aspects, legal aspects, just by considering the pollen print, which is unique for the each geographical locality. So following points can be proved to be studied by pollen grains in forensics. It can relate to a suspect to the scene of crime. We can suspect a person at the time of crime or a particular locality. It can relate an item left at the scene to a suspect, whether the item is belongs to the suspect or not, based on the biological evidence which is part of that item and part of that person. Then we can match these two things and we can confirm, we can relate. And it can relate to the foreign item which found on the scene of the crime. So the foreign item, any part of that crime locality, which is not showing the match with the pollen grains of that particular locality, we can say that we can confirm that that item or the person is not part of that locality. It has come from the item has come from some other place. And support or disprove the alibis, the reason. Sometimes you can support or we can disprove with the facts of this pollen evidence. It can search for the trace materials to their original sources, original source from where it has come. That can be confirmed with this. And there is no time bound for a pollen analysis. We can do at any time, because most of the pollen grains preserved properly, and they can retain their characters for a long period because presence of that tough layer sporopollen which is made up of. And every contact leaves a trace. Every contact leaves a trace. So this is a pollen grain. You can see here cartoon. We caught just because of this pollen grain, unnoticed pollen grain. Because of contact of that pollen grains, it left the trees, and based on that, this culprit was caught. And each geographical region produces a unique pollen print that gives a very important clues and to the exact location, and we can identify. And these pollen grains are really nature's tiny clues in solving of the many aspects, legal aspects. Of course, this is generally methodology we use. First, we identify the plants from the locality. So identify the purpose of investigation, identify the resources required there. We take the different things from the locality, and we preserve them properly. Whatever the data we collected, that should be isolated, secured, and preserved properly. And we go for analysis. So different analysis, different procedures we use, and we analyze those things, and proper documentation can be done to produce it or to present it in front of the court or in front of the legal areas. So someone is scribbling on the screen. Please don't do it. Yeah, collection of forensic samples. How to collect the forensic samples? This is the method involved. Control and evidence samples must be collected. This is mandatory. Wherever the crime scene is there, if forensic pathologist is invited to collect the forensic samples, he must opt the method, and he must go to that locality with the proper things, and he must wear the gloves. These are all whatever he used in that collection, that must be new, so that there will be no contamination. Otherwise, if you use old things, old items for collection, there may be a chance of contamination, and that could not be established proper evidence. So one should wear the proper gloves and record the scene, photograph or take sketch or take a notes exactly what things are appeared there. Everything should be sketched or photographed and noted down, and placed in a sterile containers, sealed and labeled. All the evidences collected from the scene, they must be put in a sterile containers, which is a clean, and that should be sealed and labeled it properly. On which date it was collected, that should be labeled, and place of the name also should be given, and secure and establish a chain of custody. And sampling instruments must be clean or new. Better that should be new, so that there won't be any chance of contamination. So here the forensic pathologists are working for collection of the samples at the crime locality. See how they dust and they're collecting. So these are all, this is a kit generally used by the forensic pathologist, and this is a tape, gum tapes. If sample is too meager, too minute, in that case, very small dust is there, and we cannot collect the dust properly. So in that case, we can use the cellophane tape, and this is called tape sampling. So we can use a tape sampling or containers, all these. So various equipments, and we should photograph that scene, crime scene, and we must note down all the things which are present at the scene. So what to collect, how to collect? The collection method of control samples is called a pinch method. Pinch method is nothing but it is 5 to 10 grams of material should be collected from the surrounding areas of the crime locality. So this can be correlated later with the evidence sample and that control sample. If they both matched, then we can say that we can confirm that crime has taken place there itself. Or they are not matching means crime was taken place some other place. It was taken some other place, and the body or item was shipped from that place to here. So we can confirm that. So 5 to 10 grams of material should be collected. This is called a pinch method. And 50 to 100 square meters around the crime locality, that premises, in that surroundings, that radius we should collect. 50 to 100 square meters radius is very important for collection of these control samples. And the contamination should be avoided. After collection of these pinch samples, generally 8 to 10 pinch samples are enough. So different corners we go and collect these samples, and after collection of these 8 to 10 samples, we must mix all these control samples to avoid the dominance of one pollen grains, or to get the uniformity. In establishing the fact, we must combine all those things. And so these are the bags generally we use. This is a plastic bag with the proper labeling. Here we should write all the things which are required here. And then that should be put in a plastic bag, and then that should be put in these covers and sealed properly, and handed over it for a custody. So after collection of each sample, that should be placed in that sterile bags, what you have seen there, and tightly sealed. This will ensure that no pollen contamination occurs between the time of collection and the laboratory analysis of the sample. So if the sample is dirt, what you collected, the dirt or mud is moist, once you put it in that bag, plastic bag, and once you zipper bag and you closed it, there is a chance of getting contaminated with the fungus, and the evidence sample may be destroyed. So to avoid that, once you collected that, if they are moisture is more in that samples, you put it for drying in an oven. Just slightly open it, the cover is open, and put it in an oven at low temperatures, so that the moisture will dry off. Then that can be put for a preservation. Or else, if that's not possible, you have to add alcohol. Alcoholic preservation is the best one if the sample is at low, very minute. And what evidence is we should collect from the locality, crime locality. Whenever there is invitation from the police officials or crime branch for the forensic palynologist for the collection of evidences, these are the various examples of trace evidences should be collected from the crime locality. Hair, dust and sediments, clothes of that particular person, shoes, intestine parts, vehicle, air filters, honey, coins and paper currency, illegal drugs, dry fruits, tea and coffee, furniture. If you find all these or any of these from the crime scene, immediately you should note down and you should collect these things properly and preserved. And they give you very important clues. Here you see the hair. Hair is an excellent pollen trap. This is a picture of pollen photomicrograph along with the hair taken under the scanning electron microscope. See how pollen is trapped in between the hair. So different people have different styles of hair. Hairstyles is different, and some are having the straight hair, some are having the curly hair. So straight hair, this curl hair is it traps a more amount of pollen than this straight one. And even some people use hair oil, hair tonic, and that increase the trapping nature of that hair. It traps more amount of pollen and spores while moving. And the hair can be sampled for its pollen contents by carefully washing with the detergents and warm distilled water. Once it is washed, whatever the sample collected from the crime locality, that should be washed. Yes, Ramadevi raised hand. Any doubt? If you have any doubt, I can answer, or else at the end we'll go for question hour.
Ramadevi madam, do you have any doubts?
Shall I continue?
Yes, sir. Yes, sir. Please.
Yeah. So once the hair which is collected from the crime locality, that should be washed properly with the distilled water or detergents, and that should be stored in a sterile containers and refrigerated. Otherwise, that will be contaminated with the fungi or some other things. And we can also choose not only hair, the other things which are made up of hair or fur coats, blankets, felt hats, or animal skin based products, which are excellent pollen traps, and that should be collected from the crime locality. And other thing is clothes and pollen in the atmosphere is continuously settling because of the gravitational force. It always settle down. So any person moves in the air, his clothes or hair, all these parts get trapped with the pollen grains because they're exposed to the open air, and always the pollen spores which are floating in the air because of the gravitational settlement, they fall on the people or items. So woven material, particularly left exposed to their air, becomes coated with the airborne pollen and the spores that are trapped in the fibers of the material. You can see here, this is fibers. These are fibers here, how they trap the pollen grains. Yeah, and you can also see here, this is this mark, yellow mark is nothing but it is all pollen attached to this cloth. Here also you can find the traces of the pollen grains on this cloth. And different kinds of dresses, in different ways people wear, and each dress has its own impact in trapping of the pollen and the spores. So persons, particularly people like India, we have a different cultures. As phytogeographist Boots said, vegetation of particular locality decides the climate, and climate of that particular decides the culture of that particular people. So cultural diversity always leads to the wearing of different clothes or styles, and this also gives some important clues, geographical identity, and as well as based on that whether exactly the person has come from that particular geographical locality. The pollen print also, if match, then we can confirm based on this evidence, pollen or spore evidence. And see, shoes also good for the trapping of pollen, particularly canvas shoes than these leather shoes. So these canvas shoes, while walking on the grass, the pollen of grass definitely attached to these canvas shoes, and we cannot also put these shoes for evidence. And the fibers of these canvas shoes or a rug, whatever, it holds the pollen grains. The fibers holds the pollen grains, skin cells, mold spores, dirt and dust, pet dander, bacteria, and dust mites, and all these can be found in these fibers, between the fibers. So sometimes these are all helps in the solving of the different legal aspects. So another important aspect, honey, it also plays an important role. One case was solved just by analyzing the honey sample. One person was traveling and disappeared, and someone murdered him and dumped somewhere else. Nobody knows the person from where he has come, because he's new to that place. Nobody knows. Just he was holding a little sample of honey. So it was given to the forensic pathologist, and they analyzed that honey sample, and they found that the body was come from the desert location, because the pollen which is depicted in that honey sample showing the desert plant pollen. So they went to that place and they find out that culprit based on his relatives' complaints. So honey also plays an important role. Honey is the analysis of the honey is called as a melisopelnology. Melisopelnology. We identify the pollen grains present in the honey. Generally, bees collect the honey while nectar. While collecting the nectar, they also collect the pollen and nectar mixed with the nectar. Pollen is meant for their protein source, and nectar is meant for their carbohydrate source. For that reason, deliberately they collect the pollen grains and mix that pollen in the honey samples. So after analysis, we can know the botanical source, how many plants bees visited, that we can confirm. If plants visited 30 plants, those 30 plants can be identified just by analyzing the pollen present in the honey. And we can also confirm the geographical source. Just I told you, Kashmir honey. They are claiming Kashmir honey. Whether it has come from the Kashmir or not, we can confirm just based on the honey sample analysis. If the sample consists of the rhododendron pollen or else pollen of the Crocus sativa, Kumkumpu, which exactly particularly grows in that area, Kashmir locality. If the pollen is present in that locality, then we can confirm this honey has come from the Kashmir locality. Like this, we can identify the geographical source. Based on that, we codify the honeys like Ooty honey, Coorg honey, Kashmir honey, Rangareddy honey, Khammam honey. Like this, we can codify and put it for a market. So even this codification also helps in the solving of the various aspects of the crime. And here you see these honey samples. Geographical origin is chestnut honey basically comes from the Turkey because the presence of pollen nearly 93%. If any pollen present in the honey more than 45%, then we can confirm it as a unifloral honey. If it is a mango pollen there, then we can call it as a mango honey or sapindus honey. Like this, we call based on the pollen percentage. So citrus honey comes from the again Mersin and Turkey, and clover honey from the Turkey, cotton, Adana and Turkey, sunflower, Adrian and Turkey. So these are, of course, it comes from the sunflower, also comes from other places, but along with sunflower, which other pollen, 45% is a major pollen. See here, 78% is a major pollen, and remaining pollen percent shows particular geographical location. We can confirm that geographical identity we can make. So honey also plays an important role. It gives clues. And imported dry fruits. Imported dry fruits also have the role. So dry fruits comes from the different countries. Many of the business people claim that these are all imported fruits. Have imported from the different localities. Whether exactly from that locality or not, we can identify the geographical location of all these dry fruits by just analyzing those or putting for the pollen analysis. If the pollen analysis, the pollen print exactly match with that particular geographical location, we can say that these dry fruits have been imported from that particular locality. That can be confirmed. So and teas, there are can anybody answer how many?
Sir, your microphone is mute, sir. Kindly unmute. Sir, Vijay Bhaskar, sir.
Yes. I'm taking water.
Could you please repeat the question once again?
Yeah. How many teas are available? Anybody having idea? there are 20,000 of more than 20,000 of tea available in world. Okay, so Assam tea, some teas are very special. Some teas are very special and branded and cost high. So what people do, simply they adulterate these teas with the normal leaves, the normal tea leaves or some other leaves. They mix with this and they put it for sale. If we done forensic or pathological analysis, we can confirm the origin of all these teas exactly from Assam or some other place they have come from, and even adulteration also we can find. So this is one aspect, and the other thing is coffee. Coffee also, there are various types of coffee available, and one important coffee that is very costly coffee in the world called Kopi Luwak coffee.
Please
Yes, what happened?
kindly mute your microphone. Rajesh,
Shall I stop or?
1 minute. Rajesh, can mute that participant because you are muted. Rajesh, Rajesh, kindly mute your microphone. Now you can continue, sir.
Yeah. Why they are unmuting? Yeah, please don't unmute your mic. Okay, so this is most expensive coffee in the world is a Kopi Luwak, which is brewed from the beans partially digested and defecated by the Asian palm civet. So the excreted parts of that beans of this civet used as the costliest coffee. And generally this comes from the various localities. Even people used to get it from Visakhapatnam, Vizag. That also one costliest coffee in the world. We can confirm the authenticity of these coffees. See, while digesting these beans eaten by this civet, the pollen of other plants also goes inside. But one important thing is here, the outermost layer hexane is coated with a sporopollen, made up of sporopollen, so that the biological systems cannot be digested easily, the pollen. So the pollen also mixes with this and comes out. And by analyzing these coffee beans, we can confirm the authenticity of these coffee's origin of the place. And we can avoid the adulteration of all these products. And paper currency, paper money or coins, which are particularly associated with the illegal drugs or coffee tea transactions, are a potential source for collection of forensic samples. See, majority of the people who smugglers particularly smuggle these illegal drugs from one place to other place. If they caught along with the paper currency, that paper currency also holds the pollen of those illegal drugs plant. So with that we can confirm the origin of that illegal drug. And vehicles, the dirt found on a stolen vehicle or vehicle used in a crime could link the vehicle with the scene of crime or link to the place from where it has stolen. So you see here, this is an air filter inside we use, and it traps the pollen and other parts here, so that while traveling, it also filter the air and it traps the various types of pollen, spores, bacteria, fungus, and all these comes here. So once you analyze this, the pollen print of these filters can give you the vehicle where it has come from, in which areas it was traveled. That travel history also we can know. And even the mud which is attached to the tires, the grooves of the tires. And some places, some people abandon the vehicles for at a crime locality and goes away, disappears. And after a long time, they will find out that crime scene, which was detected in the place, and any vehicle is abandoned there for a long time. Pollen is trapped here or fallen on these vehicles, and this can be put it for evidence in which season the crime was taken place, because over a period of time many other things we lose. There is a law that gradual change takes place, and that gradual change leads to the reducing of the evidences. So, but here pollen, we cannot. No one generally natural things cannot be destroyed, so that these pollen can retain for a longer period, and we can put it for evidence, and we can identify exactly in which season the crime was taken place. And other thing, important thing is the illegal drugs. We can trace out the travel history of these crude drugs or illegal drugs, from which place they have come from. Particularly the marijuana. This is cannabis sativa, locally known as ganja. They are grown and harvested, processed and shipped from the locations that gives a clues to trace out its geographical location. So generally they are harvested along with the marigold, or else what is called Hibiscus cannabinus, gongura. Okay, it exactly looks like. And other plants which are part of that location, the pollen also fall on these leaves, and once they packed it, put it for sale or smuggled to some other place, and whenever they caught, just by analyzing those samples, we can trace out their place of origin. So that's one thing. And this is a poppy, Papaver somniferum. So while extraction of Papaver somniferum, opium is made up of just tapping the parts of this Papaver somniferum, so that the latex is oozed out. This latex is powdered and put it for sale. People take this is again illegal drug, banned in many countries. So while tapping the latex, which is oozes out from these pods, can ability to trap other pollen grains which are part of that locality, so that once it is powdered, then we can analyze this powder and traced out other pollen which is part of that locality, particular locality, and we can find out the roots of these illegal drugs which have come from that particular region or particular place. And even in the case of cocaine also, the cocaine leaves are picked up and put it for dry in open areas. So while putting for dry, other pollen also fall on the leaves, and they pack it and prepare cocaine. And that cocaine, once you analyze, we can trace out the pollen of other plants which have that particular locality, and we can trace the locality from which these illegal drugs have come from. So another interesting thing which was developed by the pelnologist of the University of Surrey, what they did, generally bullets packed with the pollen could help in solving the gun crimes. Nowadays gun culture is increasing. Most of the people are using gun, and pollen has evolved an extraordinary stickiness and resilience that making it as ideal for identity tag for bullets. So what they did, these people collected pollen of this Liliaceae member and load it along with this powder charge here. They load it in the cartridge. Okay, so most of the time, if it is not coated with this pollen, generally earlier the forensic officials depends on the DNA profiling or skin cells or other biological material which is stuck to the gun cartridges here, these parts, while loading the bullets into the gun. cartridge or whatever. During that time, the DNA or fingerprints or any biological material which is attached to that, that can be used, but it has only 10% success rate, because DNA or biological material is damaged by the heat produced when the bullet is fired. So once it is filled with this powder is filled along with the pollen, this pollen, if all ammunition were marked with the pollen tags, they could help police find out when and where bullet had been bought. So each ammunition is given one certain type of pollen. So that particular batch is filled with one batch is filled with Liliaceae pollen, one batch is filled with Agaveaceae pollen, other batch is filled with particular plant pollen. Even species wise also we can segregate and we can fill and wherever crime took place, and we can confirm that. So this has come from the particular locality, and this was given to the particular person for usage, and this was misused, and the culprit will be caught. So this is a very recent technique developed by the University of Surrey, and this technique can be implemented in different ways to trace out or to find out. Even scientists have palynologists have submitted some proposals to the governments, various governments, to place some pollen grains in the paper currency, which cannot be easily traced out, and any fake currency can be traced out if doesn't have the pollen, particular pollen, which is part of that currency. So once it is the extraction of pollen and spore samples, how to extract the pollen and spore samples. Am I exceeding the time? What's the time remaining, Dr. Kavita?
Yes, sir. How much time you required, sir?
10 more minutes.
Yeah, sure, sir. You can.
Yeah, fine. Extraction of pollen and spore samples. This is the method generally people use a small faint brush and take the samples from the extractor samples from the shoes or ties, whatever the thing equipment got from the traced out from the crime locality. And this is the procedure generally we use: analysis of pollen and spores, electrolysis technique, or heavy liquid treatment, and we trace out. And this is a light micro techniques, various sophisticated techniques, equipments have evolved time to time. It depends on the modern techniques to trace out the or to get the exact marks of these pollen and spores. And I'll give you two important case studies. One is Texas murder case, which was solved by the Professor Von Bryant. And see, this was the dead body found near the major highway of in West Texas, and it was identified very late. And by the time the whole body was decomposed, and it was left with skeleton and these clothes and shoes. That's it. Nothing else. No evidence was traced out from that place. And later on, this was forensic officials collected these clothes, shoes, and also the mud samples from the surrounding samples and given to the palynologist. And palynologist analyzed and found the two pollen grains, Alnus and Shepherdia. So alder pollen is wind pollinated one, and Shepherdia pollen is insect pollinated. Of course, wind pollinated means there is a chance of coming from the other areas because of wind, but there is no chance of alder pollen or Alnus pollen from that locality, because it grows very far away, 250 miles away from the north locality of Texas. These two are existing. So actually, this is the place where murder took place, and actually the plants are growing here. So search for evidence was shifted from this locality to this locality, and finally they solved this crime. So another interesting thing is this is a shroud of Turin, which is a very worldwide attraction was taken. So shroud of Turin, also called as a holy shroud, which was used to wrap the body of Christ when he died on the cross. So after a burial, and this cloth was recovered, and that was displayed in the Turin, which is a part of Italy. Actually, Christ was died in Jerusalem, buried there, and third day he resurrected, and the cloth was left over there, and that was taken by the different people, and finally it went to the Italy, European country. So there, Turin in Italy, it was now it is displayed there. When the popes asked given this to the different forensic officials and forensic chemistry, forensic various fields worked on this. Even palyologists also worked on this, and this is the person Max Fry. He recorded 58 different taxa of pollen from this shroud, So just by using the sticky tape samplings, and 58 pollen samples, out of 58 pollen samples, 38 samples were from the Middle East region, particularly of Jerusalem, and 7 are unknown, and 10 are of Europe. These 10 of Europe are because of the that cloth is displayed in the Europe. So with that, they found that 10 pollen grains are for Europe, and remaining all from Middle East region. So these are the key pollen grains recorded from that shroud. This is Zygophyllum duonosum and Gundelia tourniforte, Cistus christicus, and Caparis aegyptia. So these are all pollen grains, and particularly these key pollen grains are only part of this Jerusalem and not part of European countries. Any European countries, you don't find this plant. So this confirms the authenticity of this shroud. It has exactly come from the Jerusalem, not part of European countries. And this was presented in the International Botanical Congress, which was held in 16th International Botanical Congress, which was held in St. Louis. That code is called as a St. Louis code. So there are many opportunities in the forensic science. One can choose as its career. There are different universities and institutions offering this course, and even Swan University is also offering a course. And one can, who is interested, and one can join for the either BSc or MSc course, and can become a forensic DNA analyst, or a forensic pathologist, or a forensic accountant, or a forensic engineer. And there are so many fields one can choose, one can opt, and gives you better opportunities, job opportunities. And every year there are notifications inviting from the people who did their master's degree or bachelor's degree in forensic science, and institutions and other private or public institutions are offering jobs for these people. So anyone interested in this, one can opt this. And thank you all for your attention. Thank you so much. And if you have any doubts, you can share with me.
Yes, sir. It was wonderful session. Now I request all the participants, if you have any doubts, you can interact with the
Shall I stop screen share?
Yes, sir. Yes, sir.
Yeah.
I request all the participants, if you have any doubts, you can ask. Or else you can also type in chat box.
Actually, I would like to present so much of cases, but cases are not important because subject is important. So I concentrated mainly on subject, how palynology works on the forensic science, and given very few two important case studies. There are so many case studies, cases are there solved by using the pollen grains. Yeah.
Any doubts? I think no more doubts, sir. Okay. So now I request Pavitra madam.
Yes, I'm there.
Now I request Pavitra madam for vote of thanks.
Okay, thank you, Kavita madam. A very good afternoon to one and all. I, Dr. Pavitra Vali Karsha, faculty in microbiology, take this opportunity to propose vote of thanks on behalf of principal, organizer, staff and students of Telangana Social Welfare Residential Degree College, LB Nagar. I thank our guest speaker, Dr. Vijay Bhaskar Reddy Garu, Assistant Professor, Paleontology Laboratory, Applied Paleontology Laboratory, Department of Botany, Usmania University, Hyderabad, for accepting our invitation and delivering an excellent talk. Sir has given a very good introduction of forensic science and its interdisciplinary relevance. Forensic paleontology, the term itself sounds to be interesting. The pollen grains or spores playing important role in forensic science and criminology. Pollen acting as evidence in crime scenes is something amazing. Sir explained very clearly about the principles and laws of forensic science. He has given a detailed description of pollen structure, their dispersion, and their role in identifying or solving a crime. Sir has highlighted about the samples to be collected and the methods to be adopted in collecting the samples from the crime scene and their importance, and also about the extraction of pollen and analysis. And he also explained about the applications of pollen, and the case studies shared are amazing. Thank you so much, sir, once again for your valuable time and sharing your knowledge in forensic paleontology with us. Thank you so much. On this occasion, I would like to thank our Honorable Secretary, Dr. R.S. Praveen Kumar Garu, IPS and Joint Secretary, Dr. Praveen Mamidala, for their leading supervision and inspiring us to do the best. I extend my gratitude to Dr. Bhanu Prasad, OSD, and Dr. Rajini Pasupuleti, Zonal Coordinator of South, for their continuous motivation. I express my special thanks to our beloved principal, Madam Dr. V. Arundhati, for encouraging and extending her continuous support in conducting this webinar. I thank Dr. D. Kavita, Head Department of Botany, and the organizer of this webinar for inviting such an eminent speaker and arranging very interesting talk on forensic paleontology. I also thank all the faculty members of our college for their cooperation in conducting this webinar. I thank Swati, Anuradha, Faculty of Computer Science Department for their technical support. Last but not least, I thank all the participants, including faculty members, research scholars, students from various streams for their attention and patient hearing. Thank you, one and all. Over to Kavita.
Thank you, Pavitra. Thank you. Once again, thank to everyone, and also thank to Vijay Bhaskar Reddy sir for spending valuable time with us. Thank you all the participants for making this webinar success. Thank you.
Thank you all.
Yes, sir.